So now it's my great pleasure to introduce our first speaker, which is Professor Olexandr Isayev. So Oles is Assistant Professor at the Department of Chemistry at Carnegie Mellon University. He hails from Ukraine and received his PhD in computational chemistry at Jackson State University. He's since worked at Case Western Reserve University and the University of North Carolina before taking up his current position. He's received numerous awards for his research, including the ACS Emerging Technology Award and awards from NVIDIA for GPU computing. So Oles, I look forward to hearing more about your work, and the floor is yours.
Thank you, Ben. Hello, everyone. It's really a pleasure to be here and share, some of the success story of our research. And the title of my talk for today is Accelerating Drug Discovery with Machine Learning and AI. So before I run out of time, let me acknowledge actually people who've done all this hard work. So members of my lab, students and postdocs, my collaborators from the Los Alamos National Laboratory and University of Florida, so a group of Adrian Royberg, a good friend of mine, partner in crime, and many of the projects, funding from the National Science Foundation, Office of Naval Research, and BASF, and some of the chemistry and synthetic work and biological measurements been done in collaboration with Inamin and ChemSpace. So two companies that actually still works and now in Ukraine and in extremely hard conditions we're actually synthesizing compounds that I'll show today. And finally, a lot of computational chemistry work been done thanks to the some of the HPC resources and agencies that provided high performance computing support. Since I'm the first speaker, so let me start from a couple of motivational and introduction slides. What you've seen is that AI and robotics and all this emerging technology has been all over the news, including the popular news, but also the scientific news. So here on the left, this is Royal Society of Chemistry news that says a brave new world of robot chemists and synthetic farms awaits. Wanted synthetic chemists, humans need not apply. So on the right, this is chemical engineering news. So this is the news outlet for the American Chemical Society. That's the rise of the smartest machines. So, we see all the hypey kind of titles. But what I'd like to start my talk is actually from this thought-provoking quote attributed by to Derek Lowe, who write amazing blog called In the Pipeline for the science translational medicine. So what Eric said, and as a pharmaceutical industry veteran, is that it is not that machines are going to replace chemists. It is that chemists who use the machines will replace those that do not. I think this is interesting, thought-provoking quote, and certainly that motivates some of our work. Now, let us start with the kind of high level. What is machine learning? And here, I think the best would go to the pioneer in this field, specifically Herbert Simon, who was a brilliant scientist, Nobel Prize laureate and also Turing Award laureate. So what Simon said is that learning, just overall learning, is any process by which system improves performance from experience. Now, when we talk about the machine learning concern, so this is specifically a computer program that automatically improves their performance through experience. And that would be, generalized definition for the machine learning. Now, this field is rapidly growing, and specifically, this very interesting review published just recently in the ChemRev that essentially described the whole zoo of AI methods. What is AI? What is domain? What is the difference between machine learning and AI? You can see on this flow chart, different types of methods and, different connectivity and types of the questions we may address. So there's many different methods. However, specifically for classes of chemistry problems, we can look at this little table, and we can map different classes of machine learning methods. For example, Bayesian probabilistic methods, or symbolist or connectionist would be neural network methods, to a type of chemical queries. And we can map naturally, types of questions we want to answer in chemistry to a most appropriate class of methods in the machine. By far, most popular and most prolific type of methods what are used in most sciences, actually, what's called supervised machinery methods. And you can generally think about it, about this magic function that we want to connect some sort of property, maybe energy, that may be, physical, chemical properties of a molecular system. And the goal of machine learning would be find this magic prediction function F, connect X's with Y's. This is typically done through what's called the process of training. So we collect a historical data of pairs, X and Y's. So we have molecules and their properties. And then the goal of supervised machine learning framework, find this magic function F. So essentially minimizing the prediction error on this training set. Now, in order to understand if it's model good or not, we typically use what's called the testing. Now we will test this function F to new examples, new molecules that model never seen before, what we call these test examples. Given X, we can predict Y and then go back to the lab or run the simulations and see how good or bad our predictions. So this is overall conceptually how the supervised machine learning framework looks like. Now, to what kind of classes we can apply it? And, for example, we can apply it to the process of, chemical process optimization, finding the best solvent for this specific process. In drug discovery, specifically finding a molecule that gives us the best receptor binding, or in organic synthetic reaction, we want to optimize catalysts to maximize yield. All of these things can be done essentially fitting magic function F, fits in molecules, give us property or quantity of interest. Now, specifically in drug discovery, it's been applied quite widely, in particular in the early stages through what's called the virtual screening. An idea is that we use this high throughput computational method to screen very large collection of molecules, millions or even billions, to prioritize several manageable number of candidates. It's called the hit molecules. And essentially what we do is we're looking for this problem as finding the needle on the haystack. Now, so our function would be, function of molecules to, for example, receptor binding or whatever the protein interest. And I think what conceptually it has been applied for many, many decades, but what new advances in machine learning allow us to do, we can flip this problem and we can do inverse. Given a property, can we find molecule X? So f of Y give us X. And essentially, instead of very inefficiently sifting through needles, we can make a model to generate the needles. And here is, where we come for a conceptually new type of ML and AI models, what's called the generative models. And mathematically, again, we have our samples denoted by X, our numeric property by Y, and essentially when we do what's called the forward or virtual screening, our model essentially models this probability of samples, probability of Y given an X property given a sample, and therefore it comes from molecules, go through molecule, through some kind of machine learning models, essentially embed those molecules in this mathematical high-dimensional space, and then looking for one with the best properties. With generative models, or in some field what's called inverse design, models inverse probability distribution of function P, X given a Y, so sample given a label, and it solves inverse problems that we want to specify desired property, embed them in the mathematical space, and then reconstruct molecules that correspond to that property. So there's two conceptually different approaches. And why would you want to use these generative models? And there are multiple reasons. So first of all, it's exciting new type of models, but also currently when you look into commercial and proprietary databases, so our biggest databases approaching billions and tens of billions of molecules. However, the mathematical estimate of the possible universe of organic drug-like molecules is going to insanely large number, 10 to the power of 60. So really, really huge numbers. Therefore, if you're looking for new drugs, current known molecules covers only small pocket of space. And therefore, maybe why we cannot find this next big concept drug, because we have this observational bias that we look only in the small little pocket of chemistry, therefore ignoring all these possibilities of new exciting chemistry. And therefore, this is where the value of generative models kicks in. Now, you probably have seen some of the flavors of these models. You've seen deepfakes and various applications of the manipulation of the images, where you can take a style of one picture and transform it to another, right? You can take, for example, glasses and add glasses to a picture of someone else, or you can take style of a language and a speech and gives a voice of a famous person, so forth. So all of that is enabled by these generative models. And again, the way how it works in pictures, we're looking to learn this probability distribution functions. And when model learn this probability distribution function, so when we sample from that functions, it will look, for example, these illustrative examples. If you look to learn, how to generate, pictures of dog, this probability distribution would learn what is meant to be, a picture of a dog. So it learns, representation of a dog in the image, the ears, the tail, and so forth. And therefore, then you can sample from that probability distribution function and get your, essentially fake images of dog. Now, more relevant example would be a chemical example, right? You can learn the probability distribution, what it's meant to be organic molecules, and then sample from it. So here, figure on the right shows you some of the examples of the sample molecules from a model we train. And to me, it looks like normal organic drug-like molecules. You get typical aromatic cycles, heterocycles, and functional groups. Now, however, molecules are not images, right? And therefore, many labs and academic groups and work in this direction where focused on molecular representations. Therefore, because again, molecules are not pixel in the images, therefore, a generative model for molecules can be done through several different representations. Some of you might be familiar and seen. So for example, you can encode two-dimensional structure of a molecule through what's called a SMILE string. So SMILE strings essentially would be a sequence of character. It's like a molecular language that in the sentence encode two-dimensional structure. It's 1D because essentially it's a string of characters, but it encodes to the structure of the molecule. And more natural for us as a chemist would be a molecular graph. So it would be one-to-one correspondence of atoms would be edges in this graph, and the chemical bonds, the atoms would be nodes in the graph, and the chemical bonds would be edges in this graph. And even more relevant would be, collection or, one three-dimensional conformer, because molecules are not flat, they're, three-dimensional objects. So all of that can be valid representation, and therefore you can generate molecules, as the SMILE string, as the 2D graph, or three-dimensional conformer. In our work, we're not only interested to, generate the molecules, because if you just give a molecule, it may not be interesting. But we want to generate a molecule with specific properties. And a few years ago, we were one of the first lab who apply reinforcement learning to optimize properties, and therefore generated these needles with the desired set of properties. Our inspiration were the AlphaGo system. And you probably have seen this in the news. It's a deep learning systems where it plays game of Go, which is arguably the most complex board games. And essentially it moves checkers on the board. And it uses reinforcement learning, how to teach system to play the game. And essentially, you know, it moves checkers on the board. It interacts with the environment, in that case, with the human, with the board. And the system looks to maximize the reward. In case of the game, you know, if you win the game, you get a positive reward. If you lose the game, it's your negative reward. Now, conceptually, you can think about that. Let us play a molecular game, right? Instead of moving checkers on the board, we will draw a two-dimensional, you know, structure of the molecules. And as a reward, we would use another ML model that would predict our useful property. Therefore, our system consists of two neural networks. One is generative, that generates a molecule, and second is predictive. So it scores the molecule for our particular property of interest, and then a crafty reward that give it back to say, hey, system, this molecule is not interested. Oh, yeah, this or it's very interested. And therefore, it uses reinforcement learning to teach this generative model to generate better and better molecule. Now, this is how it works. Imagine I have two neural networks, one generate a molecule, and second would predict, for example, a binding affinity of a molecule to a particular protein. So we generate the particular molecule, feed it into our predictive model. Predictive model says, it's inactive. Then we will use methods of reinforcement learning, in particular, what's called the policy gradient methods, to change weights of this generative model in order to increase the probability of reward. And we iterate. When you run this in cycles, eventually you generate the next molecule, it's fed to a predictive model. Now it's predictive as an active. And now we can use a positive reward. Okay, say, yes, this is a good example. Eventually, after many, many cycles, you pre-train this generative model to essentially give us only good molecules as predicted by our predictive model. So they're putatively, active molecules. And now we have our happy generative model. Now, we can look at the progression of this training as this little animation. So what this graph will show you, essentially a probability of our molecule being active. And here we use just a cutoff of 1 macromolar activity binding to a specific receptor. And the beginning of training, most of them molecules is inactive. You see probability is equal to 0. As I run our cycles of training, you see that system shifts, and now we have higher proportion of molecules being predicted as active. Now, can we put it into practical exercise? And so this is just a kind of schematic overview for the full workflow. Again, so this is our reinforcement learning system. So first, we train generative model on a large collection of organic drug-like molecules from the database called Campbell. So there's a collection of several millions of molecules being tested in various biological assays. And then our predictive model would be a little ML model that's trained for historical, measurements to a particular receptor we pick for a project. Then, we train it, generate several, thousands of molecules. And then, since we are a small academic lab, we face the dilemma, right? If you draw an orbital molecule, it's going to be very hard to synthesize it. So we did a little trick. We partner with a company called Inamin, that essentially is CRO, a company, that provides commercial service to synthesize a molecule. And we look with the very large collection of molecules at Inamin, and as soon as we generate a hit, Inamin can synthesize, we keep it. And therefore, we can save money, on the custom synthesis, and then order molecules through Inamin, and then select it, Inamin synthesize, ship them, and then we test molecules in biological assays. Now, as the several protein targets. Again, this project is done in collaboration with structural genomic consortiums. This is a non-profit organization that interested in finding inhibitors of various kinases and also chemical probes for understudied or what's called dark kinases. And we've done this exercise to several different kinases. I'll show you just example for two. And specifically, we've done it for EGFR. So this is one of the kinase proteins that implicated with cancer and inflammatory disease, and CSNK-A2. Again, this is one of these dark kinases when we have very little idea how this will work. Now, then we set an exercise. Essentially, can a robot, this AI system, beat a human? If we would be audience, I would poll you and ask how many of you believe the human will win or a robot will win. But again, unfortunately, impossible in the webinar format. But the long story short, so this is again, pipeline of experiments. Model automatically select a collection of molecules again to a certain budget we have. Then these molecules were synthesized and tested in kind of multi-stage process. So we've done enzymatic assays. So first, we've done very inexpensive single dose experiment in 10 macromolar concentration against specific proteins. When the molecules were found active, we follow up with the full, dose response, measure IC50 using, threefold serial dilutions. And finally, most promising candidates can be tested, for example, in cell-based assays, for example, things like nanopret assays. And basically, let's give you kind of in vitro, cell-based activity, and this enzymatic assays in vitro, and the cell-based assay will be in vivo. So some results for EGFR. Again, we selected 30 molecules for experimental validations. We selected 25, positive, potential hits. And just in case to see is 5 molecules were selected as the negative control. So those are molecules that system generated, but predicted as inactives. And again, in many of these exercises, I think very important to have this negative control. Out of these 30 molecules, 25 were successfully synthesized. Again, that shows you that chemistry is hard. Unfortunately, out of 25, 3 were insoluble, and therefore we were not able to run any biological assay. So eventually 22 molecules were tested for EGFR. And all 5 inactives were confirmed as inactives. Again, it's very good to have this safety check. Out of remaining 18, we have 4 compounds were found active, confirmed with the IC50s. And, this is PIC50 values. Basically, we had, around, macromolar or, a few hundred nanomolar active molecules. And that's correspond to a 27% hit rate. But EGFR, it's very well studied target. Again, there's 2 FDA-approved drugs, so it's not very interesting target per se. So this is again my second example. So this is CSNK-A2 kinase. Again, it's very understudied kinases. Again, we ordered about 50 compounds, 38 were successfully synthesized, and 7 confirmed as the active. Now you see several of them, values of measured activities. So for example, the most potent compound have 650 nanomolar IC50, and the rest have low of macromolar, and the 7th one is actually inactive. We follow up this most potent compound was also found be active in the cell-based assay, and that molecule can be again now been optimized into lead optimization phase with finding more and more potent compound, and that kind of show you that again we can work as this well-tested and understudied targets as well. So overall, was the answer if the human or a robot would win. So overall, our hit rate about 20 to 30%. Again, using fully automatic selection, no human brain involved. And this actually hit rate, it's about on par with traditional virtual screening projects, which is typically guided by expert medicinal chemist who picks molecules. And overall, that sort of gives you an idea that, again, modern AI methods, given properly trained and applied, can indeed mimic skills of the expert chemists, and can be used for this autonomous generation of new chemical entities. And we show in multiple experiments that the success rates is not a coincidence. So this is the first story that show you how ML can accelerate drug discovery. Now let me switch a gear and talk a little bit more about more basic research. Because the background in my PhD, I'm a computational chemist by training. I think there are tremendous opportunities how ML and AI can accelerate more broad computational chemistry, which would help drug discovery as well. Now, in computational chemistry, we face this dilemma that if you deal with the very large system, then typical quantum mechanical methods are very expensive, and therefore for realistic system like proteins, nucleic acids, realistic materials, if you want to do large-scale simulation, you have to rely on the force fields because your system is thousands or even millions of molecules. Therefore, you have this trade-off. So you can kind of look for this ladder of computational methods when on x-axis you have the scale of your method, and y-axis would be some sort of metric of accuracy. And therefore, if you're looking into simulate large molecular systems, you have to rely on these force fields, which are essentially linear scaling methods. However, force fields are good, but the problem with them is that you cannot systematically predict is it going to work or not for your specific systems. Therefore, the uncertainty, the error bars can be very large. Your simulation can be very good, or it can be terribly wrong. And to systematically lower those error bars, you have to account the fact that molecules are quantum objects. And therefore, you have to climb this ladder and include more and more rigorous description of quantum mechanics. So for example, density functional theory would be one of the workhorse methods. And coupled cluster theory would be a gold standard for many of the experiments, but you pay the price at 7, it's scaling as the n power of 7 with the system size. And somewhere here, for this purist, if you want the exact solution, so this is your full CI and complete basis set, this is somewhere inachievable, heaven for exact solutions. Now, this machine learning method, what you called machine learning potentials, posit to bridge the gap, right? We can use quantum mechanical data of our choice as a training data, and we can train these ML potentials that would combine, and approach accuracy of our reference, QM simulations, and be approximately, on the speed of the force field-based simulations. Now, how this will work and why this will work. So this is your quantum mechanics 101. So this is my time-dependent Schrödinger equation. If you look at this complex equation as the data scientist, you would realize that our ground state energy can be rewritten as this magic function f depending on molecular coordinates. So this is our supervised regression problem again. And what we typically do to make these models, in my labs we use neural networks. You feed molecular structure into neural networks, you predict energies. And one of the beautiful properties of neural networks, they are fully differentiable. Therefore, you can backpropagate and obtain forces. And therefore, you can do molecular dynamic simulations, you can do geometry optimization, and all these types of simulation with those computational games. Now, how this will work? I kind of think about this problem when we work with the neural networks as the building the Lego blocks, because you essentially engineer the architecture of the neural network and build this little Lego building blocks. So that would be layers of your neural network, and each block would be like a little neuron in the neural network. So over the year, we developed a number of different architectures, and this slide shows you several of those, and the reference here below. It's quite a busy slide, but let me guide you to it. So these things in yellow would be your input quantities, typically your molecular coordinates in, and energy or energy and properties out. So those are yellow. Things in blue, those are mathematical transformations. So you take your molecular coordinates. Unfortunately, Cartesian coordinates are not very good descriptor for the molecule. Therefore, you have to transform it somehow to satisfy requirements to be a molecular descriptor. So you transform Cartesian coordinates, some kind of embeddings that you feed into green blocks. Green blocks would be the blocks of the neural network. And from the simplest to complex, more complex methods, and for the rest of the lecture, I'll show you several examples and describe how this architecture works. So Anakin, or Ani for short, how we call it, is one of the first methods that's been found practically useful. And for those of you who are interested to learn more about the mathematical formulation and kind of logic, why we made certain choices, shameless plug. So this is one of the review we put a few months ago in the accounts of chemical research that describes essentially all the architecture I will feature in the rest of my talk. So again, back, one of the first and simple architecture we call ANI, or Ani Deep Neural Network Potential. And to my knowledge, that is the first example what's called transferable neural network potentials, which mean that it's applicable to many classes of organic molecules if the type of elements in V parameterized is included, and to numerous studies, what we and many other people show that for standard drug-like organic molecules, it gets you accuracy of energies within 1 or 2 kcal per mole from our reference DFT, and being also many orders of magnitude faster. And this is chemical science paper, which is original reference. Now, how this will work? So the core of that approach, it relies on what's called nearsightedness principle of chemical interactions. You've seen all this graphs in the PChem books that many types of interactions that essentially you can truncate at certain distance and ignore because essentially your interaction go to 0 at certain cutoff. And therefore comes idea of representing molecules through what's called atom-centered atomic environments, where each atom inside the molecules represents essentially a fingerprint vector in a sphere of a certain radii. So this choice of this radii would be your parameter, depending type of your application. In our case, was about 5 angstroms, and therefore this and it's centered on our atom of our choice, and therefore it encodes this atomic neighborhood within essentially 1 nanometer sphere around the atom. Therefore, your neural network would zoom into one environment at a time, and the total energy of your molecule would be sum of these atomic environments. Now, how to train these things? Again, you collect your training data, in our cases where essentially millions of small organic molecules. You pass them in, then you featureize, you transform three-dimensional Cartesian coordinates into these atom-centered environments. You pass them through the neural network. Then neural network predicts these fictitious atomic energies. You sum them up, and now you look how good or bad your total energies are with respect to your ground through quantum mechanical, compute cost gradient, errors, backpropagate and iterate, and basically train. At the simulation, or how we call inference time, you pre-train this neural network, and now you can plug it in your favorite simulation package, which fits in molecular coordinates in, gives you energy, forces, and properties. For computational chemists in the audience, again, we currently parameterize mostly biogenic elements, carbon, oxygen, nitrogen, fluorine, halogens, and sulfur. We have, more elements in progress. Our particular choice of reference quantum mechanics was, this family of DFT functional omega B97XT or omega B97MV, which is slightly modern flavor from the same family. So those are range-separated hybrid functional. And we also have neural networks that have been trained to reproduce couple cluster calculations as well. Now, I will not bore you with many different applications you can do, but just show you a couple of examples. With the respect of traditional force fields that have really hard time to describe, elements, polarizables that have, hypervalency, things with sulfur, things like that. Those are traditionally very hard to describe. Our neural network potential, gives you again here on the left, very accurate description of potential energy surface for this molecule. So these examples of running through potential energy surface by varying various dihedral angles inside the molecules. You see comparison DFT versus neural network, and gets you very accurate description with all these little kinks on the potential energy surface. The couple cluster trained neural networks, this is right on the right, is give you extremely good accuracy in gas phase reaction thermochemistry, because density functional theory is in particular approximation have a often hard time to describe, for example, reaction that deal with the conjugated systems. But neural network trained to the couple cluster can actually approach you with the accuracy of the couple cluster calculation, but a million times faster. And again, there's a reference that describes those studies in details. Now, how about drug discovery? So gas phase thermochemistry is good, but I think we are now in this time when we can really apply them in drug discovery project as well. And the state-of-the-art method would be free energy simulation, when you use molecular dynamics or various types of sampling to simulate protein ligand binding interactions through free energy perturbation or some other methods. And the typical performance of that, for well-behaved system, 1 to 2 kcal per mole for the binding free energy estimations. However, because of these deficiencies in the force fields, many cases we have this, unphysical artifacts or errors, and especially, force field have this really hard time to describing distortion conformational changes that are very important for conjugated molecules, which most of drug-like molecules are. Now, in one study, what we've done is essentially we took what is de facto the benchmark standard for the free energy simulation. This was called Fep Benchmark. So this is a reference. So we took one protein. So this is TYK2. Again, it's one of the kinases. Then the nice, kind of feature of this data set that, it has very high quality free energy, experimental binding free energy, measured for this congeneric series of molecules for every target, and therefore it became a benchmark for the accuracy of the simulation. So a figure on the left gives you an idea about the typical accuracy for this particular target. As you can see here, despite the fact that root mean square error for this target is 1 kcal per mole, you can see that there's a very large number of molecules that are outliers. And therefore the R square, essentially, for that is only 0.42. Therefore, the ranking of this ligand is not very good. Now, we don't have a neural network that can describe both ligands and protein and water yet, although we work on that. However, what we can do, we can describe only a molecule through a neural network, and the rest would be described through a standard force field. And therefore, you essentially will do hybrid ML-MM simulation. This is exactly what we did. And we ran the simulation, with the force field. And now, if we replace the part of the force field with a neural network for the ligand part. Long story short, so on the left, this is our old picture, 100% force field. On the right, if we replace the a ligand with a neural network, immediately what you see, drastic reduction of outliers. So the error, drops in half, and R square going from 0.4 to 0.86. So immediate improvement accuracy, even we describe only small portion of the systems. And it makes sense because the ligand is the hardest part, essentially, for the force fields. And it did, again, in another study that essentially done the same type of simulation, what they show, if you do the conformational sampling, so this is a particular kinase compound, that if you use a force field, this is your torsional potential energy profile for this molecule, for these two torsional angles. And if you run QM, and you will believe that quantum mechanics can give you a right answer, you see these two potential energies have nothing in common. And in contrast, if you use any potential neural network, it's very, very close. And therefore, that's one of the reasons why you can substantially improve accuracy of free energy simulations. Now, all of these features implemented in a number of our codes. It's all on GitHub. It's called the Torchani. Again, implement in many types of simulations. You can do geometry optimization, molecular dynamics, various types of thermochemistry, conformational search, and so forth. Now, in the last part of the talk, I show you one example. For the couple of years, we worked to improve, and with the idea, can we put more physics and a bit more accurate? If you remember, again, our analogy with the Lego. So this is our original architecture. And again, since we rely on this nearsightedness principles, we truncated, many type of interaction, although it works. But again, as this long-range interactions are also important for many classes of molecules. Therefore, we can improve of our original architecture. And first things, what we can do, what we can do, we can switch from prescribed formula of our atomic environments and let neural network learn it, right? Because maybe by learning it, you'll find is better. Second, we can add this missing long-range interaction, and this is done through what's often called the message pricing, where this different type of environment can exchange information through what we call in our language of messages. And this is how our second architecture was born, and we call it Aimnet. And inspiration comes from atoms and molecules neural network that add these several features, and in particular, again, we have this learnable embeddings, we have this long-range interactions, and we can also learn it in addition to one properties. We can learn to multiple different properties at a time, and all of these things are beneficial. So let me quickly show you examples. So, for example, is this message passing or iterative updates, as we call it, does it improve performance of the neural net? And the answer is yes. So the graph on the left give you various quantities and the error with respect to a DFT. And as you can see, the Aimnet gets you systematic improvement. So this show you on the right relative errors, again, for a number of different properties. And you can find, for example, optimal parameter for how many iterations you pass these messages. Since we work with small molecules, 3 was optimal, gives you a best performance. Another interesting feature since now we have a fully learnable embeddings of our features, we can study, and essentially you can do projection of this learned representation and essentially reconstruct, a little corner of the periodic table. So they show you what's called t-SNE projections of these learned vectors that corresponds to each atomic environments, and each cluster corresponds to each particular element. So you see carbon, nitrogen, oxygens. You see halogens here. Interestingly, as in periodic table, hydrogen was found to be closer to halogens, which kind of makes sense. But if you zoom in into little cluster, for example, for carbon, inside the carbon cluster, you can find different pockets of carbons with different types of substituents. And therefore, you can do a little bit, more chemical aware analysis and open this big black box of the neural networks. Finally, what I've seen so far is, we feed geometrical descriptor in and predict energy and maybe properties. What is missing, to truly, transferable description is the notion of charge and multiplicity, because you might have same geometry in different states. So our final work was extension of our Aimnet. And essentially what we did, we conditioned this neural network on a spin and multiplicity and add this extra block, which we call neural spin equilibration. And this truly allows us to do transferable molecular potentials for neutron molecules and molecules in arbitrary, charge and spin states, and therefore details in this paper. So very briefly, how this little block works. It's essentially, distribute, the spin polarized charge across the molecules. And this is an example of prototypical conjugated system that have electron donating and withdrawing group. So if you either add, attach or detach electron from this system, what DFT tells you, this is very essentially non-local interactions because of this conjugation. And you will see this behavior, which typically called spin wave behavior. So you have plus and minus phases in the spin density. So if you just use geometrical descriptor and will not tell you which specific spin multiplicity state to neural network, it will just delocalize those charges, and you will get very qualitatively wrong picture. If you apply, again, our little trick with the spin equilibration, you can approach to essentially, correct solution with the neural network. We train it again for molecules in different states, neutral, cation, radicals. You'll get, again, expected 2-3 kcal per mole accuracy. However, the beauty of that is now we can apply that not only to predict energy, but also various chemical properties. And here we come to of this little trick that because we designed this architecture now, we can use neural network and generate all those great reactivity indices. So if you remember, there is a theory called Conceptual Dft that relies various properties of partial derivatives of the energy with respect to the number of electrons. And you can compute various indices, things like Foucault indices, molecular hardness, Felicity indices, and so forth. All of these related as the partial derivatives with respect to a number of electrons to the system. Now, what this architecture allows us to do, as soon as you train it, now you can fully bypass quantum mechanics. You can change number, spin multiplicity state, compute charges, and compute all of this property, fully bypassing quantum mechanics. And it works beautifully well. So those are, several regression plots, to predict this electronegativity, hardness, and electrophilicity indices. And now we apply that to, again, in the various projects. For example, you can predict outcome of chemical reactions. We can predict regioselectivity. in the electrophilic aromatic substitution. So this example of a bromination reaction. So state-of-the-art methods would be quantum mechanics compute all of these indices, and you will get, and so in this little table would summarize you, essentially you will get about 90% accuracy to predict the outcome of this reaction. Out of the box, if you swap those quantum mechanical calculation from the descriptor from Aimnet, you can get essentially the same accuracy, but million times faster. So now this truly allows us to do high throughput access to all those chemical reactivity descriptors for practical applications. It's been already used in industrial settings. Unfortunately, I cannot tell you the company, but the company essentially use it for predicting outcomes of the coupling reactions. And again, internally in there, they use the quantum mechanics to achieve essentially 80% accuracy to predict the yield of the reactions. And swapping in expensive DFT calculation for Aimnet, they again seem exactly the same. And what they report to us, they essentially verified it prospectively, synthesizing hundreds and hundreds of new molecules with essentially 90+%. With that, let me finish that. I think right now we're really excited to see all these technologies when they converging in, and it's very exciting to see for me as a computational person that we are approaching the systems that would indeed allow us to do this fully AI-driven automatic discovery, maybe even in real time, when your AI system would give you, again, come with design of the molecule, then we can synthesize them in the robotic system, then be tested in the high throughput assay, and then inform our AI if it did a good job or not, and therefore truly, accelerating molecular discovery. And with that, let me finish and leave you with this number of links. Most of our codes on GitHub, our neural network potentials plugins, available in many commercial and open source packages, our quantum mechanical data, if you want to use it for your own projects, again, on GitHub, our codes and methods used in many academic, government, and, commercial institutions. And with that, thank you very much for your attention. I'd be happy to answer any questions.
Thank you, Oles. That was a really fascinating talk. I'm not sure if you're aware, we recently had an election here in Australia, and all the candidates were very keen to be pictured holding cute dogs. So you did very well to get some cute dog pictures in there, but now I'm wondering if they're actually real dogs or computer-generated ones we were seeing. Okay, so we've already got some great questions. So the first couple are referring to the early part of your talk. So someone was asking about what is the hit rate for your drug screening projects, and I think you gave an indication of that, 20 to 30%. 20 to 30%. Yeah, but can I follow up? How would that compare to a more old-fashioned in silico screen where you use just a docking approach or something like that to screen a very large library?
What again, maybe I'm very bad with docking. We never had any good success with just pure docking.
Yes.
So I think, if the medicinal chemist would carefully look at, then you can match it. But if you just pick top ranking compounds, it's typically the hit rate would be much lower, again, given our experience.
Yeah, great. Okay, a second question relates to how large should the training database be to generate a good predictive model? And perhaps we could ask the same for the generative models as well.
Yes, so generative models is easy because you don't need labeled data. You just need structures of molecules. You just go to big collection database, there's no problem. The predictive model is the hard part. You need at least few hundred of high quality experimental data points, desirably for the same modality. So if you have KI or IC50, so it have to be the same, more the better. And this is usually the weakest point for the generative models.
And in those sort of drug design projects that you were talking about at the start, can your predictive model be used to be predicting more than one quantity? So for example, you might have the binding affinity of the ligand to the protein, but could you have also included a prediction of solubility as well to
Yes, I kind of skip it for the interest of time, but absolutely. You typically want to do multi-objective optimization, so you want to control multiple properties. And you can craft a reward through physical, you can do solubility of targets, and then, however, again, more complex your reward is high, the harder it is to do.
Mm-hmm. That makes sense to me. We've got a lot more questions than I realized appearing on here, so let's try and get through them as quickly as we can. So Victor was asking regarding the these generative and predictive models, how are problems of adversarial networks avoided? Do you know what he means there? He's saying unreasonable inputs designed to game the network.
Yes. Again, it's a very good question. GANs, specifically general adversarial networks, are extremely hard to train. They're not very stable. Therefore, our approach is easier since we use recurrent neural network in the essentially as a generative after regressive mode, and therefore those typically much better behaved. And so again, if you send me emails, we have actually a technical paper that gets you these little tricks, what to do to improve this behavior and how to get the training easier and faster.
Mm-hmm. I'm just going to pick one of Victor's other questions. At the end, he asked, how do you deal with nuisance compounds? Do they get outputted by the system?
I see. Yes. Yes. The validity rate is not 100%. We just filter out through certain property and connectivity and little filters. And I personally think it's not a big problem. Just filter them out and then just don't care.
Mm-hmm. Okay. Johannes is asking, what's the molecule size limit for the Aimnet NSE network? And can you extrapolate it to larger systems or complex systems?
We train it to small organic molecules. We run just simulation, we run probably 1,000 atoms. And it will not work on protein and water. So there's no more miracles here. But it will work on larger, maybe microcycles, large organic molecules. Small peptides, probably.
I was interested at the end when you were talking about the free energy calculations that improving the ligand potential had a huge effect. Does that mean that in the sort of classical force fields, the ligands are the problem, that the protein is being handled quite well?
Yes, it's typically because proteins 50 years and proteins 20 amino acids. There's very little variation. And I think the proteins for field are very good, very hard to beat. But the ligands and the custom chemistry have sulfur, phosphorus, you get all this weird conjugation and effects. This is the hardest to fit to the to the functional form of the force fields.
Okay, Haibo is asking, as a molecular modeler, we were always saying electrostatics is long range. And he says, do you have any insights why a 5 angstrom cutoff works in the model rather than, the 10, 12 or more that we'd often use?
Yes. Again, I skip a lot of technical questions, but the first I show you an examples were small organic molecules, maybe to 50 atoms. So a lot of, and they're neutral, right? So and we predicted gas phase quantities. So this long range interactions is lesser effect because it's isolated one molecule in the gas phase. As soon as you start intermolecular interactions, clusters, liquids, you need long range. So it just works because it's isolated molecules. Yeah. But you do really need long range interactions if you want to do real systems.
Okay, we've got lots more questions. We'll just get to a couple more, I think. So the first one I think is easy. Are your AI and ML models available for us to use freely?
Yes, my last slide, again, send me emails, it will be on the record. It's all on GitHub, free, it's all in Python. You get examples, little tutorials, Yep.
someone is asking, can the machine learning be trained on DFT modeling? Presumably you can use whatever the best sort of QM approach is that right, for the properties of interest?
Whatever level of QM is appropriate for your simulations. There's no restriction.
Neha is asking here, how are the hit rates compared to technologies adopted by AI companies like Atomwise or in silico medicine?
Great question. I asked somebody. We don't know because they show only promotional marketing and they never give us the exact answer. So truly, we have no idea because they say it's 100 or whatever percent success. So it's impossible to estimate, unfortunately.
Mm-hmm. Okay, and I think maybe the very last question for today, I'm jumping right to near the end here, which is someone asking about the Reacts FF force field, and asking how does that compare to your models in speeding up the computational chemistry calculations?
Stay tuned. The paper will come soon. We have a fully reactive potential in the pipelines.
Great. I think for the interest of time, we're going to have to stop here. But the very last question, I think, sums it up. He asked, do you have a postdoc position opening in the near future? And to me, that...
Yes, we do. Yes.
But to me, I think that really characterizes how exciting and interesting your talk was. I'm sure you'll have lots of people interested in that position.
Yes, we have students, graduate students. Although, after Australia, you will be underwhelmed by the weather in Pittsburgh, unfortunately.
Great. Well, I think we better wrap it up there. So thanks again, Professor Isayev, for your really excellent presentation.
Thank you so much. And thank you, organizers, for putting this beautiful symposium. Thank you.